Hello everybody, welcome back to our virology series, and in previous videos we have covered the structure, shapes and classification. So today we are diving into the life cycle of the viruses, and we will understand it step by step how it is crucial for the virus to infect the host cells and how we can combat the viral infections. So let's get into the video. The virus life cycle consists of the 5 main stages that we have previously discussed as well, that is attachment, penetration, replication, assembly and release. And each stage is critical for the virus to successfully infect a host cell and produce new viral particles. The first stage is attachment, where virus recognize and bind to the specific receptors on the surface of the host cell. And this interaction is highly specific, much like a lock and key. And the viral proteins or the glycoproteins on the surface of the viruses bind to the receptor molecules on the host cell membrane. For instance, if we take an example of HIV, so it targets the CD4 receptors on helper T cells, while the influenza virus binds to sialic acid receptors on respiratory epithelial cells. And this specificity determines the host range and tissue tropism of the virus. Once the virus is attached, it proceeds to penetration stage. There are several mechanisms by which virus can either enter host cells, including direct fusion, endocytosis, or injection. If you talk about the enveloped viruses like influenza virus, it typically enters through the endocytosis, where the host cell engulfs the virus in a vesicle. And if we talk about bacteriophages, so they inject their genetic material directly into the bacterial cell and they leave their capsid outside. The third stage is replication. Once the viral genome is released into the host cell cytoplasm or nucleus, the viral then hijacks the host cell machinery to replicate its genetic material and produce viral proteins. And such examples like the herpes virus replicates their DNA in a nucleus using the host DNA polymerases, and some of the RNA viruses like coronavirus also replicates their RNA into the cytoplasm using their own RNA-dependent RNA polymerases. Then we have retroviruses like HIV, which reverse transcribe their RNA into the DNA, which then integrates into the host genome. Then we have the fourth stage, that is the assembly, where new viral genomes and proteins are assembled into a complete virus particle, or we called it as virons. And this process involves the packaging of viral nucleic acid into the protein coats, and for the envelope viruses, the acquisition of the lipid envelope. If you take the case of HIV, the viral RNA and proteins assemble at the host cell membrane, where budding occurs. On contrary, if we talk about herpes virus, so they assemble in a nucleus and then acquire their envelope from the nuclear or Golgi membrane. So now the final stage is the release, where newly formed virons exit the host cell to infect the new cells. And there are 2 main methods of release, that is lysis or budding. So the non-enveloped viruses and bacteriophages typically used the lysis or bursting method, where the host cells burst out and then release the new virons. Enveloped viruses like the influenza virus exit the cell by budding, and they acquire their envelope in the process. And this method allows the host cells to survive longer and facilitating the spread of the virus. So if we talk about each of the stage, it is a potential target for the antiviral drugs and therapeutic interventions. By disrupting any part of the cycle, we can cause the hindrance in the virus' ability to reproduce or spread. For example, entry inhibitors prevent the viruses from binding to the host cells, while reverse transcriptase inhibitors blocks the replication of retrovirus. And understanding all of these stages is a key to develop the effective antiviral treatments and vaccines. So that's all for today's class. Stay curious and see you next time. Thanks for watching.